I will start in 2-3 minutes. Right, so I'll share my slides. So this is the 8th live session of this course. Up to the last class, we had been discussing the different NMRs for proteins, and we had just looked into some 3D NMR experiments. So I will start. We had discussed the HNN experiment in the last class. I'll start with that experiment, just going through it once again. So in the HNN experiment, the initial magnetization is on the amide hydrogen of the I residue. So here you can see this is the I residue, and initially the magnetization starts from here. From here it goes to the N of the I residue. So you can see, follow the arrows. From H it goes to N. From N it goes to C alpha of the I and C alpha of I minus 1. That is the same residue and the previous residue. So from N, you can follow this green arrow. It is going to C alpha I, and the other green arrow, it is going to C alpha of I minus 1. Then from these two C alphas, it goes to the amide nitrogens. So from C alpha I, it goes to the amide nitrogen of the same residue Ni, and the amide nitrogen of the next residue Ni plus 1. Similarly, from I minus 1, it will go to the same residue I minus 1 and to the next residue, that is I. So you can follow the red arrows now. So from C alpha of I, it is going to the N of the next residue I plus 1 and back to the N of the same residue, that is I. And from C alpha of I minus 1, it is going to the N of the same residue shown by this red arrow and the N of the next residue, which is I, shown by the upper green arrow. Finally, from the three amide nitrogens, they go to the corresponding amide hydrogens. So from Ni minus 1, it goes to Hi minus 1, Ni plus 1 to Hi plus 1, and finally from Ni to Hi. So you started from Hi, you're going back to Hi. Now the three time axes, one is placed on N of I, the second time axis is placed on N, this N axis. So it will give you information about N of the same residue and the residue before and after it. and the third time axis is placed on the amide hydrogens. So this experiment we saw in the previous class was giving information about 3 consecutive residues. That's why it was often called a triple filter experiment, because it was giving you information about 3 consecutive residues in a sequence, I plus 1, I and I minus 1. But what was the problem of this experiment? The problem was that HNN does not know how to distinguish between I minus 1 and I plus 1. So you will get 3 peaks, but you will not know. So if you remember, self peak, and so suppose we have something here and you get 3 peaks. We know that the self peak will have a different sign. So these two are cross peaks. But out of these two, which one is I plus 1, which one is I minus 1? That is not possible to distinguish by the HNN experiment. So then a new triple resonance experiment or 3D NMR experiment was introduced, which is called HNCN. This starts similar to HNN. So it starts from the H of the I residue. From H, it is transferred to Ni. And here is where the difference comes in. In the previous, from Ni, it was directly getting transferred to C alpha of I and I minus 1. But in this HNCN experiment, the carbonyl is a medium of the transfer. So you are not placing any time axis on the carbonyl. So the carbonyl is not getting detected. But the transfer of magnetization from N to C alpha occurs through the carbonyl. So the carbonyl is kind of like a medium for the transfer of this magnetization. So magnetization from N first comes to the carbonyl or CO of I minus 1. And from CO, it then goes to C alpha of I minus 1. So because this experiment is using this group as a medium for transfer, so the magnetization is only getting transferred in the I minus 1 direction. Because as you see in the I plus 1 direction, there is no carbonyl between N and C alpha. So this particular experiment requires a carbonyl in order to transfer the magnetization from N to C alpha. That is why transfer in the I plus 1 direction is not allowed. So the magnetization only transfers along the I minus 1 direction. So once it comes to the C alpha, now it is like the previous experiment only. So here what was happening? C alpha was giving rise to a self peak, that is Ni, and a cross peak to the next residue, Ni plus 1. So here C alpha of I minus 1, you will have one transfer of magnetization to the same Ni minus 1, and one transfer of magnetization back to the N of the next residue. So from C alpha I minus 1 to Ni minus 1 and Ni. And from these two N's, it goes to the corresponding amide hydrogens. So Ni minus 1 to Hi minus 1 and Ni to Hi. So ultimately, the time axes are placed exactly where they were placed in the HNN experiment. One is along the N of the I residue. Another is along this axis, which is detecting N of I and I minus 1. And the third is along this axis, which is detecting the amide hydrogen of I and I minus 1. So this experiment, what it is doing, it is differentiating between I minus 1 and I plus 1. Because in this experiment, you will only get peaks in the I minus 1 direction. So suppose in here, what was shown, we had started from I. Suppose we start from I plus the amide hydrogen of I plus 1. So the first transfer will be to the N of I plus 1. Then through the carbonyl, it will transfer to the, so just I'll draw this here. Suppose we are starting from here, that is the NH of I plus 1. So from H, it will go to N of I plus 1. From N of I plus 1, it will transfer to the carbonyl, and from carbonyl, it will go to the C alpha of I. From C alpha of I, now two things will happen. It will, some part of the magnetization will go to Ni, and some part of magnetization will come back to Ni plus 1. So with the same residue and with the previous residue. And then from Ni, it will go to Hi. from Ni+1, it will go back to Hi+1 where you had started. So ultimately, you are detecting the amide hydrogen of I and the amide hydrogen of I+1. So depending on which residue you are starting from, you will be able to detect the same residue, which is the self peak, and the previous residue, that is the cross peak or sequential peak. So now here some examples are shown. In this experiment, suppose we are taking a plane along the F3 and F1 frequency axis. So you have three frequency axes corresponding to these three time axes T1, T2 and T3. Suppose we are taking a plane at which F2 is fixed. So F2 fixed means F1 and F3 is vary. In this kind of a plane, what will happen? So F1, you can see, is so this F1 is basically your N, 15N, and F3 is your amide hydrogen. So what will this plane give you? This plane will give you, for the same amide hydrogen I, you will get two peaks, I and I+1. Why is that so? Suppose we are looking at amide hydrogen. So if you look at I, the amide hydrogen that is, let's say, here. Yeah. So if you look at I, you will get the I and I+1 peak. And if you look at I-1, you will get the I-1 and I peak. So what does this mean? Suppose you're taking the cross section at a constant value of F2. So the F1, F3 plane will give you two peaks, a self peak, that is I, shown in red here, and a sequential peak with the I+1 residue, that is the next residue, that is shown in open circle here. Similarly, if you start from I-1, you will get I-1, the self peak, and you will get the peak of the next residue. In this case, it's I. Now, if you take the F1 to be constant and you look at the F2, F3 plane. So F2, F3 plane, you can see F1N, F3 is 1H. Now you will get kind of corresponding peaks. So you can see Ni, and so amide nitrogen and amide hydrogen are correlated in F2, F3. Similarly, you can see if you draw a line, F3 is your amide hydrogen, F2 is your amide nitrogen. Both are of I residue. Both amide nitrogen I, amide hydrogen I. Similarly, amide hydrogen I+1, amide nitrogen I+1. You can see amide nitrogen I+1, amide hydrogen I+1. So if you want corresponding peaks of the same residue for both amide hydrogen and nitrogen, then the cross section has to be taken of the F2, F3 plane. And if you want peaks like this, which will give an idea about the directionality of the peptide, that which direction is I+1. So normally I-1 is along the N terminal, I+1 is along the C terminal. So to give an idea of the which is N terminal, which is C terminal, then we need the F1, F3 plane where we will see cross peak only of I+1. Now, we have talked about the HNN and HNCN experiment. Now, one thing to note is that suppose, so here the above line, these are all HNN, it's written here, and the below line, this is all HNCN. Now, YXZ is a general peptide where so this is a tripeptide. YXZ are each amino acids, and these amino acids are not gly and not pro, because pro and gly are special cases. So if you have a tripeptide consisting of three amino acids, X, Y, Z, then so X is your X is I, Z is I+1, and Y is I-1. So, So we know that in HNN experiment, you will see self peak. So X, so if you connect, if you go see X and X, so that is the self peak. So the self peak is represented by the square, and when it is black, it means it's a positive peak. So we talked about positive and negative peaks before. That is just the phase of the peak. So the self peak in HNN for a general tripeptide is always positive. And the two cross peaks, so X with Y is the I minus 1, and X with Z is the I plus 1. So the two cross peaks that are happening, they will be negative. This is the general pattern that is observed when in the tripeptide none of the amino acids are pro or glycine. Now when glycine is introduced, so in case of H and CN, what will happen? You will only see the I and I plus 1. We saw before H and CN will give you I plus 1. So if you see the comparative of X, Y, X, Z, these two can be compared. These are comparative spectra. One is HNN experiment, one is HNCN experiment. So in HNCN, you will not observe this peak, Y peak you will not observe because Y is I minus 1. HNCN will only give you I and I plus 1. So you have the self peak X with X, that is positive again, and the cross peak X with Z, that is I plus 1, which is negative. Now when you have a glycine, now here the glycine is in the I position. So the self peak of glycine is shown by squared. So glycine has a very unique position of self peak. Self peak of glycine is always much more upfield shifted. So, and also you can see the sign changes. In a general tripeptide, the self peak was positive. In glycine, the self peak has become negative. So that is one difference. Another difference is that in a general tripeptide, the two sequential peaks, I minus 1 and I plus 1, was negative. When glycine is in the I position, the two sequential peaks, you can see this is the peak for Y, that is I minus 1 is negative and I plus 1 is positive. So in all the cases, the black represents positive, red represents negative, square represents self peak, and circle represents sequential peak. So you can see, again, if you relate HNN with HNCN, if you relate these two, you will see that here what is happening, you will not observe again, this will not be observed because Y is I minus 1. That is not observed. But another thing that is happening, the sign pattern is also changing. So the sign when, just one second. So basically in HNN, when you had YGZ peptide, you were able to see the two cross peaks. So the self peak is negative and the two cross peaks, one is positive, one is negative. In glycine, you will only have cross peak with the I plus 1 residue. So the cross peak is here shown by the circle, and both the cross peak and the self peak in YGZ HNCN experiment is positive. So you don't have to remember each of these patterns. Basically, what you need to remember is whenever glycine is introduced, whether it is in the I position, I minus 1 position, or I plus 1 position, the pattern of the peaks changes. That means the phase, like as I said, in the general case, the self peak was positive, cross peaks were negative. So the sign of the peaks will change whenever there is a glycine. But not only that, in glycine, normally the self peak, you can see the squared self peak of glycine with glycine is very upfield shifted. So that is another uh, special characteristic of glycine. So you can, so what I just said, pattern of signs will change if the residue is glycine. One thing you can remember is that in a HNN experiment, the self peak of all the glycines, irrespective of position, whether it is I minus 1, I or I plus 1, the self peak of all the glycine is always negative. So let us look at this where glycine is GXZ. Glycine is I minus 1. Self peak, the square is self peak of glycine. Sorry, this is the self peak of X. Self peak of glycine means it has to be glycine with glycine. So you can see this is the experiment YGZ. So gly is I residue. Glycine with glycine is red. That means it's negative. Again, GGZ, glycine with glycine is red. That is, it's negative. Now, when you have a proline, if you remember, proline is a tertiary. So proline is basically and so on. So proline amide is a tertiary amide. So, sorry, this will be N will be CO and then it will continue to the next amino acid. So this proline, there is no hydrogen. So if there is no hydrogen, amide hydrogen in the proline, then there will be no peak for proline. So whenever there is a proline in the tripeptide, one peak less is there. So compared to YXZ in H and N, there are 3 peaks. PXZ will only have 2 peaks. Similarly, you can see here, if you have PXZ, where P is in the I minus 1 position, then number of peaks don't change. Compare YXZ and PXZ. Both have one positive and one negative, one positive and one negative peak. Here, because P is in I minus 1 position, so we know I minus 1 position does not appear in H and C and spectra, so the pattern does not change. But if you have P in some other position, look at this YXP, then you will have, so compare YXZ where you have 2 peaks, YXP will have only 1 peak, which is the self peak. Because cross peak with proline I plus 1 is not possible because it does not have an amide hydrogen. So if the residue is proline, the peak is absent due to the lack of amide proton. Now, whenever there is a glycine, the peak pattern changes, and this is a checkpoint in a sequential walk along the polypeptide. So what does this mean? So we will look at that right now. Here you have a polypeptide. The sequence is given above. V is valine, I is isoleucine, G is glycine, and the number is the number residue number in the polypeptide chain. This is the H and CN spectra. So in H and CN spectra, each residue will have a self peak that is positive and a cross peak that is negative. So it will have one black and one red peak. So valine, you can see it has one black self peak, one red cross peak. When you reach the cross peak, you can move in a horizontal direction till you encounter a black peak. So this is the self peak of the next residue. Negative, you can move in the horizontal direction. Now, this brings you to the special case of glycine. Now the sequence is IGQ, where glycine is in the I position. So glycine in the I position means it is this case. So this and this will correspond. So when glycine is in the I position, both self and sequential peak are positive. So you can see both self and sequential peak are positive. When you reach the sequential peak, again, you can move in the horizontal direction. Now you have a peptide where you have glycine in the I minus 1 position. If glycine is in the I minus 1 position, then both the peaks are red. That is, they are negative. You can see both the peaks are red. So this and this correspond. This situation and this situation is corresponding. So like that, you can move along the polypeptide and it will help you find the sequence, because we know H and CN will only show sequential peak with the I plus 1 residue. So if you have if you're moving to a sequential peak, that is the I+1 residue. Then if you're moving to the next sequential peak, that is the I+2 residue, and so on. So H and CN helps you walk along the polypeptide and find out the polypeptide sequence. That is, it gives a directionality to the polypeptide. Now, whenever there is glycine, you saw that the pattern changes. Instead of one red and one black, you either have two black or two red, and this will help you. These are kind of like checkpoints in this sequential walk. So if you see this kind of pattern wherever there is a glycine, that means your assignment is correct. Now we will look at some projections of the H and CN spectra. So you have the H and CN spectra. Now suppose we take the F2, F3 plane. We saw that the F2, F3 plane will basically give you, so this is copied from the first slide. The F2, F3 plane gives you correlation of I, so F3 is 1H and F2 is 15N. So this is the F2, F3 cross section plane, and here again you have seen F2, F3. So here 1H versus 15N. This will give you relation of I with I-1 with I-1, and so on. Now, one special feature of this plane, the F2, F3 plane, is that most of the residues will cancel out. We will only see peaks for the glycine and the residue near them. I mean adjacent to them. So for example, you can see we are seeing glycine 75, then we are seeing glycine 76. Or we are seeing, suppose, glycine 47, lysine 48. So we are kind of seeing the glycine. So all the glycines that are there in the sequence, they will appear in the F2, F3 plane. The other residues are mostly cancelled out. You can see in this spectra that you see, this is the only residue which is not adjacent to a glycine. All other residues are I+1 to a glycine, glycine 10, E11. So E11 is I+1 to the glycine 10. Then glycine 47, lysine 48. So again, I+1 to the glycine. Similarly, glycine 75, glycine 76, and glycine 35 is appearing on its own because maybe the next residue is out of range. So mostly the residues that will appear are the glycines and the residues just adjacent to them. So that is one quick way to find out how many glycines there are in the polypeptide. And how will we differentiate that this is a glycine or this is a lysine? So that we already know glycines have a very significant different pattern, or as I was saying, the self peak is very upfield shifted. So whenever you see a break in the pattern, you will know that that is a glycine. Now F1, F3 plane gives you sequential connect. So F2, F3 plane is G check, that is glycine check, because all the glycines will appear very distinctly in the F2, F3 plane. Now if you have the F1, F3 plane, this will give you an idea about the sequential connectivity. So here the F1, F3 plane is shown. On the x-axis, you have the 1H of the amide protons, and on the y-axis, you have 15N. In this plane, we have already seen, we will see self-peak, that is shown by red here, and the sequential peak of I+1. So I+1, I+1, all the peaks will, just like we discussed over here, I and I+1, this kind of peak will appear in F1, F3 spectrum. This is basically an expansion of what we have seen here. So you can see this is another sequential walk. So the peptide sequence is given here. there is a leucine at first. So you have a self peak, which is black, sequential peak, which is red. Then from this sequential peak, you're moving to the next self peak of the next residue. From self peak, again, you're going to sequential peak. Now, when you come to a glycine, you see TGK. So glycine is in the middle. When glycine is in the middle, both will be black. In this case, again, you can see both are black. So again, when you move on, GKT, here glycine has moved to the I-1 position. In that case, when glycine is in the I-1 position, GXZ, both peaks will be red. Indeed, both peaks are red. So like that, you can move along the polypeptide and find out the sequential connectivities. So the F1, F3 plane helps you find out the sequential connectivities. In order to get these planes, basically you have a three-dimensional spectra. And if you take projections of the spectra along any one face, so you have a cube kind of structure. So if this is your 3D spectra, suppose you're taking projection along one plane, that will give you a 2D spectra. So like that, you can see the F2, F3 plane is a two-dimensional spectra. F1, F3 plane is a two-dimensional spectra. So depending on what you need, two-dimensional spectra might be enough to solve the sequence. So the assignment can be done very fast in the HNCN experiment in just a few hours. Now, basically, we have covered the basics of protein NMR, consisting of sequential assignments and finding out. So we have seen that how to recognize the amino acid through TOXI or COSI, and then how to find out the sequential connectivities through HNN, HNC, and other triple resonance experiments. We have also seen some example of ROSI or NOSI, which are used to find out the secondary or tertiary structure of proteins. Now we will look into protein dynamics. Now, protein is not a stationary molecule. It moves and it has many different kinds of motions. Each of these motions have a different time scale. So you can have bond vibrations where just the bond is vibrating, and this occurs at a very fast time, 10 to the power minus 12 seconds. Or you can have a side chain rotation or reorientation. So you remember the amino acids have different side chains. So the side chain can rotate in different directions, and that also occurs in a time scale from around 10 to the power minus 4 to 10 to the power minus 11. Similarly, there can be local unfolding that happens on a time scale of around milliseconds to seconds. Local unfolding means that it is unfolding only in a certain region of the protein. Then you can also have global unfolding, where the unfolding occurs throughout the protein. So the entire protein is unfolded and becomes a random coil. And this is the highest time scale of around 10 to the power 3 seconds. So up to hours time scale it can go. So there are many different kinds of motions, and each motion has different time scale. So in order to study each motion, there are different NMR experiments. So like suppose if you want to study the global unfolding, then the NH exchange experiment has to be studied. If you want to study the local unfolding, then the J values have to be calculated. Similarly, if you want to study the very fast motions like loop orientations or bond vibrations, then the T1, T2 NOE has to be studied. So there are different NMR techniques to study different molecular motions in proteins. So first we will look at the exchange between conformational states. So suppose you have A and B. you have one state where the proton is attached to H, and you have another state where the so B plus H, BH plus A. So the proton is exchanging from A to B. This is called exchange between different conformational states. Now for this, there will be two signals, because when proton is attached to A, or in the A state, it will have a different chemical shift, and in B, it will have a different chemical shift. And the intensity of these peaks will be related to the population. So if suppose this is A and this is B, and A intensity is more, that means the population of the proton being in the A state is higher. Now we have already seen this before. I'll just quickly discuss it again. When it is exchanging from A to B, if there is no such exchange, then the peaks are very clear and distinct and sharp. When there is a slow exchange of the proton going from A to B, then the peaks slowly start becoming broader. That is, the chemical shift range or width, peak width, becomes larger. When the rate, so as the rate of exchange is slowly, you can see from bottom to top, the exchange rate is slowly increasing. And then the peaks are slowly merging. This is the intermediate exchange rate, where you can distinguish that there are two peaks, but they are merging here. Coalescence is the point where you can no longer distinguish two peaks, and there is only one chemical shift value in the middle. So if you come here, there is only one delta value. That is the coalescence point. And as the exchange becomes even faster, the merged peak starts becoming sharper and sharper. So these are the different exchange. So as the exchange rate increases, the the uh, distinct peaks disappear and a merged peak is obtained. Now, there are different factors that can affect exchange. If you increase the temperature, the rate will increase. And then in different solvents or on addition of ligands, the rate can be affected. Basically, the exchange rate is often defined. So if the exchange rate is less than the frequency difference of A and B. So this is the frequency difference. Just 1 second. This is your omega A minus omega B. If your exchange rate is much lesser than omega A minus omega B, it will be this kind of condition. And if it is greater, it will be this kind of condition. There are different equations to calculate the exchange rate. So exchange rate defined by K. There are different equations to calculate the exchange rate at different conditions. So we are not going to go into the details of that. Just you can remember that this is the case that multiple signals will be seen when the exchange is slow, and only one signal will be seen when the exchange is fast. So we have HD exchange experiments, often known as HX experiments. These are basically, you can see HN exchange. It is in the seconds to hours time scale, your minutes to hours time scale, because actually it is more than second time scale. So the minute to hour time scale dynamics, like the global unfolding, even some parts of local unfolding, or the chemical kinetics, aromatic ring flips, these can be studied by the HD exchange or HX experiments. In this, what happens? You have a NH amide proton and you have D2O. Now the D will exchange with the hydrogen, so you will have ND and HDO. Now deuterium, you know, is NMR inactive, so the H signal will vanish. So as the You won't see the H signal here because we are applying solvent suppression. If you remember, there are different techniques of solvent suppression. So you will not observe the signal of HDO. So the H signal will completely vanish as NH becomes ND. Now you have a protein that is shown here. So the blue balls are hydrogen and the red balls are deuterium. Initially, all are H. Now you are adding D2O and you're allowing time to exchange. At one point, you will see that some hydrogens have been replaced by deuterium, and initially only the hydrogens on the surface will exchange with the deuterium. You can see these two red spheres mean that the hydrogens in these two locations have been replaced by deuterium, and these are at the surface. Similarly, the other places where deuterium has replaced hydrogen are all on the surface, because the surface hydrogens will exchange rapidly. As you allow more and more time to go, the hydrogens on the interior will also start exchanging, and you can see more and more of these red balls are there. So more and more hydrogen has been replaced by deuterium. So in order for this to happen, normally what happens is that the NH folded. So this is the folded protein represented by fold. In that case, initially all were hydrogen, this blue balls. Then the folded state is in equilibrium with the unfolded state. And when the unfolded state is reached, then only the hydrogen is accessible for exchange, and it exchanges with D2O to become ND. So that is why the portions on the surface will exchange much more rapidly, because they are already exposed to the D2O. But the portions in the interior of the protein, which are very well folded into secondary structures like helices or beta sheets, they will first have to unfold in order to be exposed to the D2O, so that the hydrogen is accessible for exchange, and that is why it takes time for the interior protons to be replaced by deuterium. Now here you have a 1H15N HSQC shown, and from top to bottom it is increasing time. So top is when time is 0, that is initially, then you have 9 minutes, 63 minutes and 133 minutes. You can see there are so many peaks in the first case, which is at the start of the experiment when all the amide protons are present. So each peak corresponds to one amide proton. So basically one amino acid, if we don't consider amino acids with having amide protons in their side chains, then each peak corresponds to one amino acid, and there are so many amide protons initially. As time goes, the number of peaks start decreasing, because more and more of the amide protons are getting replaced by deuterium. So as it is replacing, that peak is disappearing, because deuterium is NMR inactive. So as you allow time to pass, the number of peaks will decrease. And this rate of disappearance of the peaks is connected with local or global unfolding events. As I discussed earlier, in order to exchange the interior hydrogens, the protein has to unfold. So depending on the rate of local or global unfolding, the rate of exchange of the amide hydrogen will deuterium will change. So this is the hydrogen deuterium exchange. Basically, again, the experiment shown has been copied here. Here you have a graph shown on the x-axis. Basically, it is showing that the exchange rate, the x-axis is basically the intensity loss. So observed NH intensity loss can be fit to a simple exponential curve in order to measure the exchange rate. So in the x-axis, you have time in minutes, and in y-axis, you have the intensity, NH intensity, that is slowly reducing. Suppose you have this innermost graph that is there, where the intensity is suddenly and very rapidly dropping. That is a case where so basically what is plotted here is three amino acids. Three amino acids have been chosen: valine, isoleucine, and asparagine. In these three amino acids, the decrease in the intensity of the peak is plotted with respect to time. Basically, as time goes, the peak that is innermost here, the intensity decreases most rapidly. That means this amino acid, the amide hydrogen, is being replaced by deuterium most rapidly. So this amino acid must be on the surface of the protein, and that is why the exchange is so rapid. Similarly, if you look at the outermost curve, you can see the rate of drop in intensity is much slower. So the exchange of the amide proton with deuterium is much slower. So this amino acid must be in the interior of the protein, because if it is in the interior, it has to temporarily unfold through local unfolding events or even global unfolding events, so that it can be exposed to the D2O and exchange with deuterium. And that takes a lot of time, because we saw that the local or global unfolding time scale is quite large. That is why this amino acid, which is exchanging very slowly, must be in the interior of the protein. So you can see there is a very clear variation in the exchange rate of the three residues. The intensity drop like this can be plotted as in an exponential function as mentioned. So there can be one exponential component as in the first case here, or it can be summation of two exponential components. When will there be two exponential components? When there are two different exchange processes. So the curves will be exponential in nature, but depending on whether there is one exchange process or two exchange process or more, the kind of function, whether there is one exponential component or two exponential component, that will change. Now, here you have more details of the experiment. So here another curve is shown. The residue number is in the x-axis, and in the y-axis, you have log of the exchange rate. You have a secondary structure here. So you have a helix. So basically, as expected, majority of the NH that exhibit slow exchange rates are located in secondary structures. So basically what was done in a protein, like here you have plotted the exchange rate of three amino acids. Here exchange rates of all the amino acids in the protein are plotted, and then it is seen that where the secondary structures are lying, that is, which residues of the amino acids are forming secondary structures. You can see the helix one here is formed from around residue 1 or 2 here up to residue, say 7 or so. Then in this amino acid portion, residue portion, you have a turn. One thing you can see that wherever the secondary structure is there, so the secondary structure is here to here, basically here to here, you can see the exchange rates are low. So secondary structure is there, exchange rate is low. Why? Because whenever there's a secondary structure, we know that the protein is very well folded. So all the amide hydrogens will be hidden in the interior of the protein in the secondary structure. So they must unfold locally or globally so that it can be exposed to D2O and exchanged. And that takes a long time. So whenever you have a secondary structure, the exchange rate will be low. Similarly, you can see helix 2 here. The exchange rate here is low. Helix 3 and 4, again, exchange rate is low. And the fast exchange regions, they are mainly located in the N and C terminal. So you can imagine this structure. The end of the protein, the N or C terminal, is generally exposed to the surface. So they can exchange very fast. So the exchange rate in the N and C terminal is quite high. And where else it is high? Between two secondary structures. So you have a helix and you have a turn. So connecting two, suppose you have a helix, then you will have something, and then say you have a sheet or a turn. So this portion that is connecting two secondary structures, also called as loop, this is exposed to your solvent. And so the exchange rate is high. You can see, imagine there is a loop connecting the helix and the turn. The exchange rate is high here. Similarly, another loop connecting these helices, exchange rate is high. So whenever there is a turn connecting, or loop rather, connecting two secondary structures, the possibility of getting exposed to the solvent is high, and so the exchange rate is also high. Now, in the exchange process, we'll see what is happening. So initially, suppose you have AH, in this case it's NH, and a base is added. So you have a deuterated base is added. So OD minus you have. This OD minus takes the hydrogen away from A. So you have A plus and HOD. HOD is your solvent. Instead of H2O, it is deuterated water. So HOD. Then this A minus, then you add some acid in the medium. The A minus will take a deuterium from the acid and become AD. And D2O will be there in the solvent. So acid or base, if you have, it will speed up the exchange process of AH becoming AD. Now we will look at how this is happening. So here you have a protein and you have a local unfolding. Let us first look at the horizontal direction. In the horizontal direction, so we are focusing at one hydrogen that is shown here. Initially in the protein, the hydrogen is in the interior. It is not exposed outside. Now, a local unfolding is happening where only this region where the hydrogen is there is getting unfolded. So now the hydrogen is exposed to the solvent. This is called the opening rate, and the reverse reaction is called the closing rate. So kind of equilibrium exists between the native structure of the protein and the locally unfolded structure. And from this, once it is unfolded locally, the hydrogen can exchange with deuterium. And this rate is called as KCH. You can consider it as the rate of changing or something. So hydrogen being replaced by deuterium. So this can only happen after the local unfolding. The other way this can happen is by global unfolding. So the entire protein unfolds and becomes a random coil. Now the hydrogen is definitely exposed to the solvent. And again, you can have an exchange rate where the hydrogen is being replaced by deuterium. So for hydrogen deuterium exchange, either local unfolding or global unfolding is essential. Now, the exchange that can happen, there can be two possibilities. One is that the exchange rate is much greater than the closing rate. So the exchange rate is this one, going from hydrogen to deuterium. If this process is much faster than the reverse process, then once the protein unfolds, immediately it will exchange. It will not go back to the folded protein. So this equilibrium will get pushed in the forward direction. That is one possibility written as EX1. The other possibility is when the reverse reaction is much faster than the exchange reaction. So in that case, this exchange will not happen only. The protein will always tend to remain in the folded position. That is called the EX2. So the rate that is observed is a mixture of these different opening rate, closing rate, and so on. You can just remember this particular equation. So if you derive the kinetics, you can find this equation. K observed of the exchange rate is K opening, the forward reaction of the local unfolding, into K exchange divided by K closing, the backward reaction, plus K exchange. Now, if you have the EX1 condition where the K closing is much less than K exchange. If K closing is much less, you can ignore this term, and K exchange will cancel out. You will have only K opening. So K observed is K opening rate. And in the EX2 position, if K closing is much greater than the exchange rate, if K closing is much greater, then you can ignore this term and you will have K observed as K opening into KCH divided by K closing. What is this ratio? This ratio is the K equilibrium. You know, K equilibrium is the ratio of the rate constants of the forward and backward reaction. So this ratio is the K equilibrium of the protein. So from the K equilibrium, you will have an idea of the protein stability. So through unfolding experiments, you can measure the protein stability. So as I discussed in the previous slide, either adding an acid or adding a base will speed up the rate of the exchange. So you can see when the pH is low, like 0 or 1, the log of the exchange is higher. Or if the pH is high, like if you have a base 9 or 10, then also the log of the exchange is much higher. There is one point where the exchange rate is slowest, that is around pH 3. You see, if you drop a perpendicular, pH around 3, the exchange rate is lowest. At this low pH of 3, where the exchange rate is slowest, different unfolding experiments can be done. So suppose you have a protein and you're adding urea. Urea is a denaturant. That means as you increase the urea concentration, the protein will unfold from its native state into a random coil. So initially, the concentration of urea is 0, protein is completely folded. You are gradually increasing the concentration of urea. When it crosses, say around 4.1, 4.2, the unfolding begins. As you increase the concentration of urea from 4 to around 7.6 or so, unfolding happens. So this region between the dotted lines is where you have a mixture of the folded and unfolded proteins. Increasing beyond 7.8 molar or so of urea, you will only have the unfolded protein. So this is called a unfolding curve. So in the y-axis, basically the fluorescence intensity is plotted of a residue like tryptophan or something which has fluorescence. So depending on whether it is in the folded or unfolded state, the fluorescence intensity will be different and you will get this kind of a sigmoidal curve. You can also plot the urea concentration versus the delta G, that is the free energy of the folding or unfolding event. You can see when the protein is folded, then the delta G is high. That means it's very stable. So this delta G is actually folding minus unfolding. So the signs are opposite. But high value of this delta G implies a very stable conformation. So the folded value is very high. As you increase the urea concentration, gradually you have a mixture of folded and unfolded conformations, and eventually the protein is completely unfolded. As the protein unfolds, the delta G decreases till it reaches a minimum when the protein is completely unfolded. So these experiments are generally done in a pH of 3 because we want to study the experiment, like we want to study the uh, experiment at the rate at which when the exchange rate is slowest. So these are different folding experiments. Now we will there are so basically why we are doing this is that we need to understand when the like at what concentration of urea the protein will unfold and at what rate it will unfold, because these unfolding and folding experiments we will be using for our NMR. And how we will do that, we'll just look at it in the next slide. So suppose you have a fully unfolded protein by adding a high concentration of denaturant like urea. So you have urea 8 molar. We have already seen when urea is greater than 7.8 or so, the protein is completely unfolded. So you're starting with 8 molar urea. So the protein is definitely completely unfolded. So if it is unfolded, all the hydrogens are replaced by deuterium. So the protein is completely deuterated. Now you are slowly initiating the folding process by adding water at low pH. So when you add water, the deuteriums will exchange with water slowly, and uh, so we will that is what is supposed to happen. But we keep the just 1 second. Normally, if you add water, the deuterium should exchange with water. But here we are using low pH. We know at low pH exchange rate is very low. So here, by keeping low pH, we are not allowing the deuterium to exchange. But we are diluting the urea by adding water, 20-fold dilution. So from 8 molar, it is becoming half 4 molar. And from 4 molar, it is becoming 1/10, that is 0.4 molar. 0.4 molar will be somewhere around here where the protein is completely folded. So urea is being diluted by adding water till it reaches a concentration of around 0.4 molar. At this concentration, the protein is completely folded. But because you have kept the pH low, although the protein is folded, it is still completely deuterated because low pH is not allowing the deuterium to exchange with hydrogen. So initial time is 0. The time after the protein is completely deuterated, suppose that is TF. At TF, you are increasing pH. As you are increasing pH, the system will start exchanging. So you are increasing pH from 3, exchange rate is increasing. So as you are increasing the pH, the deuteriums will start replacing with hydrogen. You can see initially the deuteriums on the surface, this deuterium, they are the ones getting exchanged because they are completely exposed to the H2O. This is called a so as the deuteriums are replaced by hydrogens, you are studying the NMR. So initially there were no peaks in the NMR because all were deuterated. As the deuterium is being replaced by hydrogen, you are studying the NMR. So this is called the pulse label. Suppose, so at TF, you are starting, at TF, you are increasing the pH. So at TF, the exchange process starts. Now, suppose we stop the exchange process at time TP by again lowering the pH. So we again bring the pH down to 3 and stop the exchange process. So only some of the deuteriums have been replaced by hydrogens. So normally what would happen is that suppose all the peaks are appearing together. So you are having such a crowded spectra and you cannot assign the spectra because it's so crowded. But if something like this happens, we are controlling TP. So the total time of exchange is TP minus TF because we are starting the exchange as TF by increasing pH and we are stopping the exchange at TP by lowering pH. So we can control TP minus TF such that there are different extents of exchange happening. So the number of peaks will also be reduced in the HSQC, and that will help you assign the spectra better. So by giving different time periods of TP minus TF, we will get different kinds of spectra, and by kind of overlapping the different spectra, we will be able to assign the entire protein. There is another way to monitor the protein folding, which I'll just discuss in 1 minute. Right. There is another way to monitor protein folding, and this is called the folding funnel or energy landscape. So basically, the top along the y-axis, you have energy, and along the other axis, along the z-axis, you have energy, And along the x and y axis, you have different coordinates. So when the energy is highest, it is the unfolded state. So you can see the edge of the funnel shown here. These are all different unfolded states. As you go in the interior of the funnel, the energy slowly reduces until the lowest energy point is the native protein, that is the folded protein. So during protein folding, many different pathways can be taken. Suppose there is a pathway that comes from here, then again energy increases, then decreases until it reaches here. That can be one pathway. Another pathway can be much shorter. It can just go directly here. So protein folding can have multiple different pathways. And this is called, there are multiple folding pathways possible. And the starting point can also be different. There are so many possible unfolded states. But generally, the native folded state is only one. Now, suppose we have a protein with 100 amino acids. Each amino acid has 2 degrees of freedom, the phi and chi angles, which we have already discussed before. So the total number of degrees of freedom in the protein of 100 amino acids is 2 to the power 100. So suppose the system has to scan through so many degrees of freedom in order to find what is the lowest energy conformation, then the time is too high. It is impossible for the protein to scan through all the degrees of freedom. Normally, the protein folds in much smaller time scales, like seconds to hours, compared to if the protein had to scan through all 2 to the power 100 conformations, it would have taken some time, like unimaginable amount of infinite amount of time. But the fact that in reality proteins fold in much smaller time scales of seconds to hours, that is called the Levinthal's paradox. And why that happens, that as you go down the funnel, the number of degrees of freedom reduce. So as you go down the funnel, you can see the unfolded, I'll just discard this. So the unfolded state can occupy any of these conformations. As you go down the funnel, the number of conformations it can occupy decrease. So the degrees of freedom decrease as you go down the funnel. This is helping you reduce the amount of time it takes for protein folding. Now, another thing you can see, again, I'll discard this. There are some places in the funnel where again the energy becomes high. So the energy is becoming low, again it's becoming high, again low, again high. Now, there are some places where this happens, and these places are basically called, so this is the global minima. These places can be called local minima and maxima. Sometimes what can happen is that suppose the protein is unfolding, it comes to a local minima. Now the local maxima after that is so high that the protein does not have enough energy to cross this barrier. So the protein is getting stuck in this local minima. Then it is called a misfolded state. Now of this folding funnel that you have, is shown here, and HSQC is shown at different concentrations of urea. So initially you have 8 molar urea, that is the protein is completely unfolded. So this spectra is referring to this unfolded, just 1 second. Right. So this is the 8 molar urea condition. So the protein is completely unfolded. So it is referring to this region where the protein is unfolded. As you are reducing the concentration of urea, you can see 7 molar, 6 molar, 5 molar, so on. The chemical shift, intensity, relaxation properties, everything is changing. until at 0 molar urea, the protein is completely folded. So this folded native state is referring to this region where you normally generally have only one conformation. So what you can see is that throughout the folding process, when the protein is completely unfolded, the chemical shift dispersion along the proton axis is very small. It's only around 8 to 8.6 ppm. But when the protein is completely folded, the chemical shift dispersion is much larger. It is 7 to 10 ppm. So that is one major change that is happening in the HSQC spectra. The 15N dispersion also increases. So up to 3 molar, it is like 110 to 125. But around 2 molar, it becomes 100 to 134. So the 15N range also increases, but the 1H range increase is much more noticeable. So there are different structural properties in the denatured state. So if the dispersion is varying so much, that is, in the unfolded, the dispersion is so low of the chemical shift, and in the folded, there is such a high dispersion. That means there must be different structural properties in the states. In order to study this, there is something called the secondary chemical shift, or delta S. Delta S is defined as the delta that we are observing in the NMR minus the delta random coil. So if you have a random coil orientation, so basically for each amino acid, this random coil delta has been assigned based on different statistical studies. That is, if the amino acid is in a random coil orientation, it will have a delta RC. And delta observed is the delta that you are observing through NMR. The difference between these are said to be delta S, that is the secondary structure chemical shift. Now, for a particular secondary structure, there are certain signs that are expected of the delta S. So you, in amino acid, you can have C alpha, H alpha, or the carbonyl carbon. If you have an alpha helix, the delta S of C alpha and CO is positive and H alpha is negative. For beta sheet, the signs are opposite. So basically, when delta S is adopting this kind of sign, you can predict that the protein has a propensity to obtain that secondary structure. Now you have a folding experiment here, where initially you are starting with 8 molar urea. 8 molar urea means that the protein is completely unfolded. In this case, you can see what is plotted here. The delta S of H alpha is plotted. So delta S of H alpha is showing that most of the delta S H alpha is positive in 8 molar urea. If delta S H alpha is positive, that means it has a propensity to adopt beta sheet. So in the unfolded random coil state, most of the amino acids have a propensity to adopt a beta sheet conformation, shown by this black bar here. As the urea concentration is decreasing, the positive values are reducing. You can see at 7 molar, positive values are less. 6 molar, it is even lesser. In 6 molar, what you can see that some places the delta S of H alpha is becoming negative. And negative delta S H alpha means alpha helix. So you can see this region where the delta S H alpha is negative has a propensity to adopt the alpha helical secondary structure, shown by this tube here. Then as you are going to 5 molar, there are 3 stretches of residues where the delta S H alpha is negative. So in these places, the alpha helical propensity is there. At 3 molar urea, what you can see that there is no particular structural propensity. Most of the residues, the delta S of H alpha is near 0. That means mostly wants to adopt a random coil conformation. It does not want to adopt any secondary conformation at around 3 molar urea. So what is the importance of this? Basically, so suppose initially you have a 8 molar urea. So the protein, again, so I have basically copied this into the next slide. So at 8 molar urea, we saw that most of the delta S H alpha is positive. That means it has a propensity to adopt beta sheet. At 8 molar urea, the protein structure, the unfolded structure is plotted, and these indicate beta sheet. So you can see this kind of structure that is shown. Basically, you can imagine this as one, and so sheets like this. Sorry, I'll just. So you can imagine this as one side of the sheet, this as another. So at 8 molar urea, most of the residues have a propensity to adopt beta sheet structure, which is shown here. Now, 8 molar urea means the protein is completely unfolded, so it is a very dynamic state. As you are reducing the urea concentration from 8 molar, you're going to 7 molar, what you are seeing is that the dynamics is getting reduced. This is shown by the change in color from blue to pink. It means that the protein is not that much dynamic anymore. Some structural contacts, van der Waals interaction, etc. are happening. Then when you go to 6 molar urea, you can see in 6 molar urea, a small section of residues are adopting a negative delta S. Negative delta S means they have a propensity to form a helix. So at 6 molar urea, this tube, that is the helix, is forming. And the dynamics, as always, is reducing. As you go to 5 molar urea, there are 3 stretches of amino acid where you have 3 helices forming, shown here. At 4 molar urea, there are 2 helices forming, again shown here. Now, when you go to 3 molar urea, 3 molar urea, most of the delta S are very close to 0. That means they don't have any particular propensity to adopt either alpha helix or beta sheet. They just want to stay in random coil orientation. That is what is shown here in 3 molar urea, where all the propensities are vanishing. So you can see when there was a beta sheet propensity, this kind of structure was shown. When there was an alpha helix propensity, this tube-like structure is shown. When there is no propensity at all, it is shown like this, which is a random coil. Here, what happens is that only when it is becoming a random coil like this, it can fold into a native structure. Because you can see now that it has no particular propensity to either adopt beta sheet or random coil, it can now fold into the native structure. So as you further reduce the urea concentration from the structure which had no propensity to adopt any structure, it is slowly becoming like the folded state. So you can see folded state, there are multiple beta sheets here, which are indicated by these arrows, and there are 2 small helices, one very small helix and one slightly longer helix. Eventually, as you reach the zero molar state, the protein is completely folded. So basically, as it is going through the folding process, it is forming certain kinetically favored structures, like here beta sheet, here alpha helix, and beta sheet, and so on. All these kinetically favorable structures with the non-native contacts will have to be removed to get a structure which has no propensity to form either alpha helix or beta sheet. Only then the protein can fold into the native folded state. So here you have an example of a protein, HIV protease. You have the initial unfolded state. So this is the folding funnel shown, where the rim of the folding funnel is the unfolded state and the bottom is the folded state. So initially, there is a certain preference. So as you can see, initially, there is a certain propensity, that is Firstly, at 8 molar, all had beta sheet propensity. At 5 molar, 3 small regions had alpha helix propensity. The other regions had beta sheet propensity, and so on. So initially, before it reaches this state where there are no propensity, initially there are certain non-native contacts which give the protein a certain propensity or preference. That is shown by the different colors here. As you are initiating the folding by reducing the urea concentration, the folding preference is changing. Here also you can see, as the folding is initiated by reducing the urea concentration, the preferences are changing. From all beta sheet, it is going to 1 helix beta sheet, 3 helix, then beta sheet. So the folding propensities are changing. And basically, whenever this is changing, the portion of the protein adjacent to it is also changing. So suppose you have this green, imagine the green is a propensity to form a certain, so initial preference were purple and black, but the new preference that is forming are this green and blue. We can imagine the green is the propensity to form, say, alpha helix, blue is the propensity to form turn. One thing you can see, that when a particular portion of the protein is folding, the neighboring residues will also fold. And this is called cooperativity. So when a particular section of the protein has, suppose, this region, the color has changed, meaning that it has a certain other folding propensity. It is also influencing regions that are close to it in space. So it is also influencing this region. Similarly, if this region has a certain propensity, it will also influence this region. It will also influence this region. This is called cooperativity. So far we have been talking about small proteins. Now we will look at large protein assemblies. Now, large protein assemblies, basically the correlation time is very high. What was correlation time, or tau c? If you remember, it was the time for the molecule to tumble, kind of a tumbling motion of the molecules. So large correlation time indicates large size of the molecule. Here you have a graph where in the y-axis you have, in the x-axis you have spectrometer frequency in megahertz, and in the y-axis you have the line width for the protein signal. So again, by line width it means, so this range is your line width delta nu. So line width is along the y-axis. You can, and here certain numbers are there. These numbers are the correlation times in nanosecond. You can see when the correlation time, you can see that when the correlation time is small, like 20 nanosecond or 60 nanoseconds, the line width is also small, 10 or 15 hertz. But when the correlation time is very large, that is 320 nanoseconds. So for a very large protein or protein assembly, the correlation time will be very large, around 320 nanoseconds. The line width is also very large. It is more than 40 or so hertz. So when the line width is so large, that is a problem. You can see what is happening in HSQC spectra. Basically, in large protein assemblies, correlation time is large. And if the correlation time is large, you can see the 320 nanosecond correlation time is shown here. Then you have line widths which are so large. So in that case, a modified HSQC experiment needs to be done. So basically you have for a NH peak, in a NH spectra, you will see two comps. So this is basically this NH peak. The fine structure is being shown. The fine structure will have four components. in the 4 components, if you take the frequency difference between these, you will get the J value, or the frequency difference between these, you will get another J value. Each of the 4 peaks will have different intensities. If you decouple, so this is on coupling, you will observe 4 components. If you decouple, only one peak will appear somewhere in the, all these peaks will go if you decouple the spectra, and one peak will appear somewhere in the center, which is this one. And after decoupling, if you if you do the Trozy experiment, which is the transverse relaxation optimized spectroscopy, then out of the 4 components, whichever the component is which is the sharpest, that is the component that you can isolate by Trozy. So in the Trozy experiment, we won't go into the details of the experiment. From all the peaks, you will only see the peak which is sharpest, that is line broadening is minimum. So while studying large protein assemblies, because the line width is high due to the large tumbling time, the peaks become very broad. Then the Trozy experiment is done to choose that particular peak which is sharpest, which will help us resolve the spectra better. So you can see here, here the COSY is shown on the top and the Trozy is shown at the bottom. You can see in COSY the peaks are merging, they are spread out. Because of the high line width, the peaks are not distinct. You cannot resolve the spectra. And the intensity is also low. Here 3 particular amino acids are shown. Intensity is also low. When you are doing Trozy, the peaks are, you're only choosing the sharpest. So each peak will have 4 components in the fine structure. You are only choosing that component which is the sharpest. And so in Trozy, you can see the peak is much better resolved. So now you can easily assign the protein. Also, the intensity of the peak is much higher. You can see that. So both higher resolution and higher intensity. So it is much easier to count the number of peaks in Trozy compared to COSY, which is shown above. And also the intensity is higher. So the intensity can also be calculated. So Trozy is basically a modified NMR experiment, which is mostly used when the size of the protein is very large or you have a protein aggregate. There is another experiment. It's called CRYPT or CRINEPT. Basically, what happens in that is that suppose you have an aggregate like this. So this is the free protein, which has a molecular weight of only 72 kilodalton. But when it is binding to another protein called SR1, the molecular weight greatly increases to 472 kilodalton. In this case, what happens? So tau C will also increase accordingly. Now the problem is that even large molecular weight or large tumbling time can be resolved by Trozy. But when you have this kind of binding of one protein to another protein forming a large protein aggregate, the Trozy signal is disappearing. So the association process cannot be studied by Trozy because the moment it is binding, you can see there are so many Trozy signals in the free protein, but the moment it is binding to SR1, most of the Trozy signals are disappearing. So for this kind of high molecular weight complexes, if you want to study the association process, Trozy is coupled with another experiment called CRYPT or CRINEPT. In this experiment, again, we don't need to go into the details. You can just remember that the number of peaks are retained. You can see that even after binding to the SR1, if you are doing CRYPT Trozy, the number of peaks are retained. So you can resolve the spectra and assign the peaks even upon binding. So this is another modification of 2D NMR in order to study large protein aggregates, especially the binding process. Now there is something called self-association. We will look at the process of endocytosis. Endocytosis is basically, you imagine you have a cell, and there is a component that like a compound that needs to go inside the cell. So how this happens is that there are receptors on the cell surface. You can see these Y kind of things are the receptors. The compound that needs to go inside the cell binds to the receptors, and then the cell membrane kind of forms a small vesicle, and this vesicle is called the endosome, and through that uh, through the endosome the compound can reach inside the cell. This is called internalization, and the process is called endocytosis. Because it is a specific receptor ligand binding, only the ligand that needs to go inside the cell will go inside. Now, how this process happens is that when this endosome is forming, so this is the cell membrane, it will form a kind of endosome like this, and then this will break away and go inside the cell. So this kind of endosome, so this is the endosome, and this is the neck that is forming. There is a certain kind of protein called dynamin at the neck, which consists of 864 amino acids. The dynamin has different uh, uh, domains. We need to go, don't need to go into that, but there is one domain called the GED domain that helps the dynamin to self-assemble. Once the dynamin protein self-assembles, it can form a long rope-like structure. So the dynamin protein itself is not that large. It's 864 amino acids. But once it self-assembles due to the GED domain, it can form a long rope-like structure, and it will wrap around the neck of this budding endosome. Eventually, there will be GTP hydrolysis, which squeezes the neck so that the endosome breaks away and goes inside the cell. So the way this happens is by the self-association of dynamin. So here what is shown is basically the dynamin, many dynamin proteins self-associated together. If you look at the CD, there is a helical signature. TEM experiment electron microscopy gives you an idea about the particle size. So dynamin is associating into many, self-assembling into a large rope-like structure. But if you want an idea of the particle size, the TEM can give you information of that. Now, the problem is that in this kind of self-association, the number of peaks observed in the HSQC is very small. Only 25 peaks will be seen. And mostly these will be the charged amino acid residue or the ones at the surface. So if you have a large particle, the HSQC will only be able to show the peaks of the protons exposed at the surface. These peaks are so these peaks are coming that are seen in the HSQC. They are coming from certain section of the protein that is flexible, which, like I said, is exposed to the surface. The rest of the protein you will not be able to see through HSQC. It will have a very slow tumbling time because it's a huge molecule, and it will large tumbling time means a very large line width, and so no signal. So that is the problem of this kind of self-associated large molecules. So how can we work with that? Basically, we have a large molecule here, the protein aggregate of the dynamin. It can be dissociated into the monomers, that is, it can be unfolded, and then the NMR spectra can be recorded. So you can see for the aggregate, the NMR spectra is completely like very few peaks are seen. The peaks that are seen, they are merging. It cannot be resolved. But if you unfold this, you will get the monomer. And in the monomer, you can see so many more peaks can be seen. Because the monomer is a small molecule, the tumbling time is small, and the line width is small. So the resolution is much higher, and the peaks will not merge, and you will be able to resolve the peaks and assign the spectra. Now, after it is unfolded, again you can slowly change the conditions so that the aggregate is again formed. This is called the reconstitution. So suppose this is monomer. So this is like 8 molar urea when it is completely dissociated. Then slowly you are reducing the urea concentration 6 molar, 3 molar, and ultimately at 0 molar urea, again the protein aggregation is formed. So why you are doing this is that as the protein is getting reconstituted, that is from monomer, the aggregate is again forming. If you monitor the HSQC at different stages of this association process, it will give you an idea as to which residues are associating in what order. So the sequential order of the association can be derived. And during this association also, there is obviously folding, formation of secondary structures. That also can be studied by HSQC if you take the HSQC at different stages of the reconstitution process. So here basically of the GED protein aggregate, a DMSO titration is done. Basically, like we studied that urea is a denaturant, DMSO is also a denaturant. In the presence of DMSO, the assembly is completely dissociated. So when you have 100% DMSO, you can see that when you have 100% DMSO, the assembly is completely dissociated. So you only have the monomer. So in 100% DMSO, only the monomer is present. So you can see many peaks, well-resolved peaks. You can assign the spectrum. Slowly, the DMSO concentration is reduced. As the DMSO concentration is reduced, from monomer, the aggregation is starting. So as the aggregation is starting, the peaks will start to disappear. At 50% DMSO, the aggregation has completely formed, and so you can see the number of peaks are very less, because we already saw that in aggregate very less peaks will be seen. So here basically what they have shown is that the residues that are shown in black here are the ones that are observed even when the aggregate is formed. So these are the residues that remain flexible even in the associated state. So only the peaks of these residues will be observed in the 50% DMSO aggregate condition. Because even when the aggregate is, so suppose there is an aggregate formed and there is a tail that is hanging out of the aggregate that is flexible. This tail is the one that is formed by these residues. Because this is visible even when the aggregate is formed. The other residues are the ones which are basically, as you decrease the DMSO concentration, the aggregate is forming and the peaks of the other residues which are inside the aggregate are disappearing. So this kind of experiment is called a DMSO titration experiment. So in this experiment, basically 100% to 50% DMSO, you're looking at that experiment now. At 100% DMSO, mostly it is dissociated into monomers. It is a very flexible portion of the molecule. So you can see these are flexible portions of the molecule. It is this is the condition in 100% DMSO when it is only monomer. And if you look at the secondary structural propensities of the C alpha, you can see it is mostly positive. Positive C alpha means alpha helix. So at 100% DMSO, some stretch there is alpha helical propensities. Now, as you are going to say 90% DMSO, 80% DMSO, what is happening is that in some regions, the peaks have completely disappeared. These are the regions where the aggregation is happening. Because when the aggregation is happening, the tumbling time is increasing. And if the tumbling time is increasing, the line width is so large that you won't be able to see the peak. So in this region, the peak has disappeared, means it has gone into an associated state. As you decrease the DMSO concentration, more and more peaks are disappearing. So more and more peaks are getting formed into the aggregate. You can see that in over here as well. This is the transverse relaxation rate. As the aggregation is forming, more and more peaks of the transverse relaxation rate are disappearing. That is, they are not observed in the NMR because they have formed part of the aggregate. And here it is shown in the that what is happening to the structure. You can see initially the flexible portion is the uh, so this I said is the flexible portion that is shown in black here, which hangs in a tail or something even when the aggregate is formed. So that you can see it is retained. This upper part that is there, it is there even after the aggregate is formed. So the NMR of this, so the peaks in this flexible portion, they will be visible no matter what the DMSO concentration is, because they are not forming part of the aggregate. The rest of the peaks, they are slowly getting incorporated inside the, so this cube is the associated state. So as the DMSO concentration is decreasing, the other peaks are getting incorporated into the associated state. So slowly as the DMSO concentration reaches 50%, the entire monomer identity will be lost, and only the flexible tail that is hanging out, that will show NMR peaks. And the rest of the peptide will be part of the aggregate, so the peaks will disappear. So this is how the DMSO titration can give an idea about the folding process in very large protein self-associations. So you can again see here, you have, just 1 second. You can see here, you can have the non-native structures which are transiently aggregating, and then you have the native aggregate that is forming. So how this is happening, how this kind of aggregation happens is that there can be electrostatic interactions. So imagine two molecules that are aggregating. So you have electrostatic potentials are shown. This is a lysine which will have a positive charge, aspartic acid which will have a negative charge. So positive negative interactions. Again, glutamic acid with a negative charge, lysine with a positive charge, positive negative interactions. So complementarity of electrostatic interactions can help in the association. And association can happen step by step till eventually, see, this is the net aggregate structure where you have a large cylindrical rope-like structure, and on either side of the cylindrical aggregate, there are certain flexible portions hanging out. These flexible portions are the ones where you will be able to see the NMR irrespective of the DMSO conditions, because whether it is not part of the aggregate, it is a folded region, it is a flexible region. So the NMR of these flexible tails will always be visible. But the NMR of the regions incorporated inside the rope form part of an aggregate. So they will have a very large tumbling time, and so they will have very large line width, and you will not be able to see these signals. This rope-like structure that is formed is wrapping around the neck of the endosome or vesicle. That is how the dynamine molecule self-aggregates. It wraps in this rope-like structure that is formed, it wraps around the neck of the vesicle, and GTP hydrolysis gives an energy which results in kind of squeezing the neck and releasing the endosome inside the cell. And as discussed earlier, the GED domain is responsible for this self-assembly. So you can see here, so how are the dynamics and structure important for biological function? So here you have a dynein light chain protein, or DLC8, which is a trafficking protein. This structure shown here, it is a trafficking protein. So it carries a cargo molecule from one side of the microtube to the other. Now, when this protein is in a monomer state at pH 3, it is non-functional. But when it forms a dimer around pH 7, so you can see this two blue uh, stalk kind of things are the dimer. When the dimer is formed, at that time it is the functional molecule. It forms at pH 7, and the dimer is the one that can bind to the cargo molecule and carry the molecule from one end to another end. So what is the advantage of this is that um, as you change the pH, the dynamism is changing. So you can control that. So suppose you are maintaining pH 7, then the dynein molecule is in the dimeric form and it can carry the cargo. Now suppose you have carried the cargo from one place to another place. Now you need to release the cargo molecule. Then you can lower the pH at so at pH 3, the dimer will dissociate into the monomer. And the monomer is the non-functional form. It can no longer bind to the cargo, so it will release the cargo molecule. So by controlling the pH, you can control the association or dissociation and thus allow the molecule to bind to the cargo or release the cargo as required. So that is how the structure and the dynamics is important for the biological function. So in this dynamine, you are observing 4 specific amino acids. You can see a glycine, serine, aspartic, and isoleucine. It is shown by this square here. At pH 7, a dimer is there. And so pH 7, this is the free dynein molecule. When the dynein molecule is binding to the peptide, that is the cargo molecule, there is a structural change. You can see the peak that was initially in the square region is moving to this circular region. So there is a structural change on binding of the cargo molecule to the dynein molecule. Then suppose you are lowering the pH. At pH 6, some peaks shift back to the same position that they occupied in the absence of cargo. So in the absence of cargo, the peaks were occupying this squared position. When the cargo is there, the peaks move to the circular position. Then on lowering the pH, some of the peaks will go back to the square position and some will remain where they were in this circular position. So if that happens, the binding efficiency is reduced. So on lowering the pH, the binding efficiency is reduced. So basically, you are kind of seeing a mixed NMR. You are seeing the NMR of both the dimer, free dimer, and the dimer plus cargo aggregate. So as at pH 6, some of the molecules are free and some are binding to the cargo. If you lower the pH even further, eventually at pH 3, as you have seen, the cargo will be completely released and you will only have the free dimer. So the pH can act as a switch for trafficking of the cargo from one place to another. So that is basically, this is another way of using NMR to study the structural changes in the molecule and find out that at what structure or what pH the molecule will best bind, or at what pH the molecule will dissociate. So by controlling that, we can control that when the cargo is binding to the molecule and when the cargo is getting released from the molecule. So this is another application of NMR in large protein aggregates. So now we'll quickly go through some questions, which are basically the assignment problems of week 7. The first question asked, what is the chemical shift range of backbone amide protons? So you can just remember this picture. So this will help you remember the different ranges. The backbone amide basically ranges from around, you can see 6.5 or so to 10.5. That is the backbone amide, and that is option A. So option A is the correct answer. Backbone amide basically refers to this NH, and so on. So they will be very much downfield shifted. They are the most downfield shifted, in fact, and they appear along 6.5 to 10.5, which is option A. Then you have another question. Which among the following residues does not have side chain proton resonances in the range 6 ppm to 7.5 ppm? Now, again, we will look at the previous range of chemical shifts. So you have 4 amino acids mentioned: asparagine, lysine, valine, and glutamine. Now, we will look at this, that side chain proton resonances. So we are only looking at the side chain right now. So asparagine side chain has a CONH2 group. Lysine side chain has a NH2 group. Glutamine side chain also has a CONH2 group. So there is side chain NH. So side chain NH is occurring around 6 to 7.5 ppm. So this range that has been given is for side chain NH occurring along 6 to 7.5 ppm. And this will be observed in asparagine, lysine, and glutamine. The only place where it will not be observed is valine, because valine does not have any side chain NH. So the correct answer is C, that is valine. Then it is asking, which of the following dihedral angles of a peptide can be obtained from 1H NMR spectra? Now there is something called the Karplus equation. Basically, we will look at the J of the coupling of this amide hydrogen to the C alpha hydrogen. We are calling this as JHH prime. If this is plotted along the y-axis, and in the x-axis you have the phi dihedral angle. Phi, what is phi? Phi is from C prime N C alpha C prime. So between these 4 atoms, the dihedral angle is your phi. So there is an equation called the Karplus equation that correlates the J value to the phi torsional angle. So the J value that you will get from 1H NMR will help you obtain the phi torsional angle, that is option A. So A is the correct answer. Then you have a question. Which of the following is correct? Now we will look at it one by one. NOSI is useful only for peptides of 1 kilodalton molecular weight. That is wrong, because NOSI is useful even for large proteins. NOSI does not depend, NOSI basically, it will give you an idea for much larger proteins also. So option A is incorrect. Option C is telling you TOXI aids in spin system identification and gives correlation with neighboring residue in peptides. This second part is incorrect. TOXI only helps in spin system identification. Correlation with neighboring residues cannot be obtained from TOXI, because TOXI only is TOXI or COSI. Just one second. Yeah. TOXI will not give you correlation with the neighboring residues in peptides. So that is your option C is incorrect. So option B is correct. And why is that? Option B is telling you HSQC gives the fingerprint of peptides or proteins. So now if you look at a peptide, each amino acid will have one NH proton. If we are assuming, if we are leaving out the amino acids which have backbone or which have side chain NH, then each amino acid will have one NH proton. So in the 15N versus 1H, that is the amide proton, and 15N HSQC experiment, you will see one peak for each amino acid. So it is kind of like the fingerprint of the protein, because each peak will correspond to one amino acid. So from the HSQC, you can find out the primary structure of the protein. That is, what are the amino acids present in the protein. So each protein will have a different HSQC. And so HSQC is often said to give the fingerprint of the peptide or protein. That is option B. Then you have a question that is, which experiment is more useful for assignment of an intrinsically disordered protein? Now, one thing about intrinsically disordered protein we saw in the previous class is that when you denature them, the 1H chemical shift dispersion is very much reduced. So if you look at the HIV protease, the amide proton shift dispersion is lost on unfolding. The amide proton shift here was 7 to 9.5. Here it's so small, somewhere between here and here. The C alpha dispersion is also similarly lost. So if we do the HNCA experiment, it is very difficult to resolve because HNCA will include the C alpha, and the C alpha dispersion is small, so the resolution is very difficult. Better than that is the HNN experiment, where the N dispersion is used twice because the N dispersion is retained. You can see between folded and unfolded, N dispersion in both is more or less the same, around 110 to 130. So the dispersion of chemical shifts of nitrogen is retained in intrinsically disordered protein. So a good experiment is one which will use more of the nitrogen channels. So in both HNCA and HNCO, there is only one nitrogen channel, but in HNN, there are two nitrogen channels. So B is the correct answer for intrinsically disordered proteins. Then you have a question asking, which nucleus among the following nuclei is a starting point for magnetization transfer in a 3D HNCA NMR experiment? If you remember HNCA, you start from the amide proton. Magnetization is transferred to the amide nitrogen. From amide nitrogen, it goes to C alpha of the same residue and C alpha of the previous residue. And then from C alpha, it again goes back to the amide nitrogen and the amide hydrogen. So the starting point is always this amide hydrogen, that is 1H. So the correct answer is B. Then you have a similar question for 3D HNCO experiment. It is asking HNCO correlates chemical shift of what? So HNCO, it's similar to HNCA experiment, except in HNCA, from HNCO, what happens? From amide proton, it goes to amide nitrogen. From amide nitrogen, it goes to the carbonyl carbon of the previous residue. And again, from carbonyl carbon, it goes back to the amide nitrogen and back to the amide hydrogen. So it is correlating the H of, if we just consider this as I, this as I minus 1. So the HNCO is correlating H of I, N of I, and CO of I minus 1. That is option C. So option C is the correct answer. Then it is asking, how many backbone amide cross peaks will you see in the HSQC spectra of the following peptide? If you look at the peptide, basically, it's talking about backbone amide cross peaks. So if you look at the peptide, we know that between two amino acids, there is one backbone amide. So how many amino acids are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 amino acids. So if you just look at the pairs of, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. There are 11 pairs that you can see here. But if you remember if you draw out the thing, so the peptide is drawn here. We will count the number of backbone amides. 1, 2, because there is a proline. So we counted 12, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1 second, 11. We counted 11. I'll just discard this. So, as I was saying, there will be one amide between two amino acids. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 is amide hydrogens should be there. But you have to see that one of the amino acids is proline, which will not have a amide hydrogen. So 11 minus 1 is 10. And the correct answer is B, that is 10. Then it is asking, compared to the well-folded protein, backbone chemical shift range of disordered protein is usually, so we just saw a few questions ago, HIV protease is an example of an intrinsically disordered protein. The backbone chemical shift of the disordered protein, what happens to it? It becomes smaller. This is the amide hydrogen, which is part of the backbone, and you can see the dispersion becomes smaller. Here it was 7 to around 9.5. Here the dispersion is around, say, 8.6 to 8.2 to 8.5 or something like that. Much smaller dispersion. So the correct answer is A. Finally, you have one question that is saying, tertiary structure of proteins can be determined mainly by using what? So if you remember the previous class, tertiary structure is about how the protein is folding as a whole, the global folding of the protein. So if we have a protein where the sequential connectivities have already been established, now suppose the protein is folding in this manner, then we will see nosy between A and D. If the protein is folding in this manner, we will see nosy between C and D. And if the protein is folding in this manner, we will see nosy between B and C. So depending on the nosy, it will help us find out the tertiary structure. So which is basically nosy gives you the inter-residue distance restraints. So the correct answer is D. That is, the distance restraints between residues found from nosy helps you find the tertiary structure of proteins. So that is all the slides. So basically, I have covered all the lectures of week 8 and have discussed the solutions to the assignment problems of week 7. The next class will be in this coming Monday, and where I will go through week 9. So that's all for today. As always, I'll upload the recording and the slides.